Great. Hello, everyone. I'm Teemu Laajala here on behalf of our team DenverFINRISKHacky, and I come from Finland. My main affiliation is in University of Helsinki with the prostate cancer research group over there. And in my talk, I'll be going through our participation structure, which wasn't probably the most typical way to participate in a DREAM challenge. I'll be also outlining the model structure and a little bit of our rationale behind it, why we ended up doing what we did, as well as briefly, Rebecca already showcased the performance, but I'll just reiterate on it, how we performed there. And this is my third time at a podium in a DREAM conference talking about a top performing model, 3 times participated thus far. And so I'll be doing some reflections to other DREAMs and connecting it maybe to other talks that were given today, hopefully. So I was actually in Vegas last year in the RSGDREAM 2022, when I had discussed with my junior colleagues in Denver that I had given presentations on my thoughts on how to approach a certain kind of machine learning task or survival modeling in particular, and they had asked me to, hey, could you come over and do a workshop or a hackathon for us? And yeah, well, so I was in Vegas, and I thought, okay, I'll pop by Denver, and you can see in the picture Lily, Varsha, myself, and Mike, and Kaylin behind the camera. And I had picked up from the DREAM newsletters, FINRISK DREAM challenge, and I'm not a heart failure researcher or a microbiome researcher, so I hadn't heard of the study before, but the Finn part in it stood out, obviously. I'm a native Finn, so obviously I was very interested in the data set immediately. But also it gave me a good excuse to start learning microbiome analysis. So I brought out a bunch of workshop material from others, how they processed microbiome data in R and so forth. But I thought that I could also share insights to my junior colleagues regarding especially survival analysis, where you'd have the right censoring and so forth. So the DFH, DenverFINRISKHacky, is really just Denver, FINRISK, and Hackathon combined. However, how it went with us 4 is that we had our hackathon on November, late November 2022. I brought my laptop to Denver, and we had you can see that there's kind of this is a screenshot of our GitHub commits, so basically chunks of code, as well as other materials. So you see the hackathon in late November, and you see a high bump there. And in the bimodal distribution, you can then see in late December, January, you see a crunch with myself. I'm not sure how I can pinpoint, but thanks. So you can see myself sweating over the data in January, with the second bimodal part coming in with basically we had finished the hackathon back in Denver, but I felt that the modeling that we did back there wasn't sufficient, and I wanted to give it a better crunch, really put together something that made more sense. And also we briefly touched on the topic of singularity containers, and one of the issues was that we wanted to submit our first model back in Denver, but in my Windows computer, I couldn't get the singularity containers to work. It's no problem. I'm interested in learning it, but I just couldn't get it back to work. So I had to wait until I got back to Finland and back to my desktop, where I would set up a virtual machine with Ubuntu on it, and there I could get it to work. So I had to go through some loops to get it working. So that's part of the reason why we didn't submit until January. So what we did during the hackathon was basically I tried to give my junior colleagues an overview into what kind of survival modeling they could do. And for this, there were 2 main emphases. Cox regression models, very popular, especially given when you combine them with regularization in this kind of a task where you have a large quantity of features and you need to eliminate some of those out. And we also touched on survival random forests as an alternative, but ultimately we ended up with Cox regression, probably because I was more familiar with it and more comfortable with it and the implementations available for it. We also did literature searches, a little bit different maybe from what was mentioned before. We didn't really find consistent things from the literature in regards to heart failure and microbiome. There was maybe one that I would highlight, the dysbiosis being ratio of Bacteroides and Firmicutes, but other than that, we couldn't really find anything that we could generalize across the papers. It was really just one species over there, another species in another paper, and it felt a little uncomfortable, but we wanted to incorporate those anyway, but we felt a little uncertain about it since they didn't generalize across the papers. We ended up doing a modular structure. So we built a hierarchical modeling structure where first we'd have modules where the granularity of the data would be focusing on a single thing like clinical variables or alpha indices and their combinations, and or alpha diversity indices, beta indices, and microbiome relative abundances, and so forth. And these would be subjected to regularization. Then the modules that would survive that would get, again, regularized and combined together. So we'll go through it step by step. So back in Denver, we'd basically have access to the synthetic data, and we could know what the data looks like. Immediately, we noticed from the synthetic data that the clinical variables were very dominant, like, well, what I could also deduce based on my limited knowledge of heart failure, like age and disease burden and such. But we managed to kind of get a grasp on how the data would be processed, but we didn't really get to submit a model and get a real result out of it yet because we couldn't get the singularity containers to work. But we were brainstorming and hoping to submit. So we came up with multiple modules. I like to do, when I'm modeling with Cox regression, I like to do nonlinear combinations of different variables. In this case, we had age, sex, and BMI that were really obvious ones that really dominated the predictions. But I also took a lot of transformations and combinations of these clinical variables and created modules also based on those. Then we had alpha diversity metrics, beta diversity metrics as their own modules, relative abundances across taxa. So again, this was my first time tackling microbiome, so I didn't really even know what level of taxa I should be working on. Would it be species, genuses, phylum, family, kingdom? I had no clue. So we were actually incorporating all of these different levels. And then we had literature-driven variables that we could pick up. And the last one was we combined clinical features with family and phylum level relative abundances. We binarized age based on whether or not a person was above 35 or not, if they were female or male, and then BMI being above or below 25. So that would give us 2 to the power 3 strata, and we would combine that with the different family and phylum relative abundances. So trying to combine clinical data with the microbiome. And then these would be subjected in the first phase to the regularization. So cross-validation would, each of these modules would contain a large quantity of candidate features. Some of them would already get eliminated at this phase. Like, for example, we give it the right censored survival response. But for example, in this schematic, the beta diversity module would get totally eliminated already at this phase. The others would still survive the process, but they would be very, very thin on the different variables that remained. While, for example, we can notice already that the age, BMI, and so forth, the clinical variables were still very dominant. And then we would put together these modules. And in there, we did notice that the clinical variables were dominating the final features. The one that did survive through our kind of hierarchical regularization process was the one where we combined clinical features with microbiome. So in addition to that, something that I came up was that we performed multi-seeded cross-validation just to try and reduce the amount of randomness in the data. For example, I was suspecting that due to the relatively small number of events in the data, the binning and such could also have an effect. So we would multi-seed it and then average across those different seeds to get a more robust prediction. And well, Rebecca already mentioned us and SB2. We managed to be in so-called top performers and proceeded to the post-challenge analysis. The final model, unfortunately, I have to say that it does get dominated by the clinical variables. I mentioned the modules before, and here are the different 10 cross-validation seeds. The one where I combined clinical features with microbiome did get through, but across the seeds, it's not a very consistent finding. So I think what we managed to capture well is nonlinear interactions with the clinical variables. Here you can see gender, age, and BMI combined, and with the contours, the level of risk increasing. And as you can see, it's a nonlinear kind of a heat map, if you will. And something that I would highlight for our team's performance is that I believe there's a lot of untapped potential in the microbiome that we didn't get to capture yet. So looking back at the challenge in hindsight, the other team, SB2, they had, like Rebecca mentioned, they had a clustering approach. I believe it was co-abundances. So if I could do this all over again, I would definitely go for something like that. We used pretty standard stuff, basically because I was still learning microbiome analysis, relative abundances, literature-driven markers, alpha beta diversities and such, and they were pretty much eliminated just brutally when they were not combined with clinical variables. So thinking about what I could, if I was thinking about developing new methods, I'd say, for example, there could be some kind of what I would consider gene set analysis to gene expression analysis in microbiome. So for example, with the clustering approach that SB2 did, they're bringing together multiple, I forgot what level of taxa they worked on, but family, okay, yeah, phyla, okay. Yeah, so definitely there's room for kind of method development here, I think. And I see it kind of like what gene set analysis did to gene expression analysis. Instead of individual genes, we think about pathways. So we would here think about different species, different genuses or phyla or family working together or against each other and giving us better models. But during a DREAM, especially with this kind of model to data structure with synthetic data, it's not possible to develop this out of the blue during the challenge itself. So I promise to reflect back on previous DREAMs. So I've been part of 3 DREAMs. The first one is the prostate cancer DREAM challenge back in, I believe it ran in 2015. And it had a data to model approach. So there were multiple very well standardized randomized clinical trials, and we were predicting overall survival. So we were provided with data sets, and I actually didn't have to go and look for any additional data sets. Then the results that I was presenting in Vegas before going to Denver was about the anti-PD-1 response, immuno-oncological treatment in clinical lung cancer. That was a model to data, but there we could submit things as Docker containers. So perhaps a little bit lower threshold there for participants. What they provided was synthetic data with no signal. So in contrast to what was done in this FINRISK DREAM challenge, they didn't have any signal. They just gave the matrices basically in the right format and told us that, hey, this is what the data will be in terms of structure, but you'll have to figure out a way to build a reasonable model. However, we were lucky enough to identify publicly available data sets that, well, while they didn't address lung cancer, they were mainly melanoma and some other like pioneering immuno-oncological data sets. So we could identify those and teach models and test hypotheses on those. So that was a big advantage. So when we come to FINRISK, we are then faced with model to data. And like Rebecca mentioned, the singularity containers, I personally had some problems with it during my Denver stay, but I did manage to get it to work on a virtual machine. So, well, not sure what's the pros and cons there, but at least I had some difficulties there, and I wasn't the only one. The synthetic data and that having signal is a nice idea. And we've heard some talks during this conference about generating synthetic data, and it's really an art form in its own. And I think what we, for example, during the hackathon noticed was the clinical variables dominating. And we really didn't get to practice on the microbiome that much. And we kind of felt uncertain whether or not it was worth the time to go into the synthetic microbiome data. And we weren't able to find publicly available data sets that would have addressed this question, which in a way also highlights the fact how unique cohort this is. So this is obviously not something that the organizers would have to do. So a take-home message. So I wanted to do a workshop. I wanted to do something for my junior colleagues. A DREAM challenge being the setting for it is a neat idea, but its practical implementation is very difficult. So you want to think about it very well through. The synthetic data, time management, singularity all created its own challenges. Regularized Cox regression, which is a very well established methodology, performed well. Both of the top performing teams utilized it in one way or another. And I think there's many interesting venues in microbiome research. For example, these kind of like, well, what I call gene set analysis as an analogy. Colonies are synergistic or antagonistic relative abundances working together or against each other. So the model to data, it does pose some design limitations and challenges to us participants. And there's definitely a lot of things to think about when you're organizing new DREAM challenges, and I'm sure a lot was learned already during here. Perhaps more model feedback could be provided or something. I know that, for example, more submissions are typically allowed in these cases, but it's a little bit, doesn't account for everything if there's no publicly available data sets, for example. So thank you for your time. There's a preprint out, and I'd be happy to take questions.
I think we have time for one or maybe two questions. So if anyone has one. Okay. So you talked a little bit about your initial searches of the microbiome literature and being kind of slightly frustrated by the heterogeneity there. How much do you feel that kind of understanding the domain of one of the required data types in the model was useful for creating a real model?
Well,
or a valid model?
Sure, yeah. I mean, domain knowledge is definitely a huge factor in these challenges. And if I look back into the other challenges, which were cancer challenges, we had MDs that were oncologists. So they could provide us with a lot of insight, while the colleagues I had in our team weren't also either familiar with the microbiome. So, for example, the fact that we didn't know what taxa level to work on, that was like one very basic question, I would presume, but I had no clue on how to approach that. So just as kind of try to be on the safe side, we try to incorporate all of them, which is probably not the right thing to do, but yeah, definitely could have used some experienced microbiome researchers on our team.